people present in the talk. I think if you have any questions, you can either type in or you can just speak out loud because it's a very small, informal group, and Professor Bittu will be tailoring their talk according to the questions raised. And I think a short introduction about Professor would help our participants prepare questions as well. So Professor Kaveri Rajaraman is an associate professor of biology and psychology and is currently the head of psychology department at Ashoka University. They received a PhD from Harvard University in neuroscience. Yeah. And their work is on the evolution of neural and behavioral system of communication, the neuroethology of temporal patterns, recognition in insects, and quantitative and economic cognition more broadly in zebrafish, dogs, and humans. So, yeah, this is their research area, and please feel free to ask any questions if you have. Or, Professor, maybe you can just start by casually talking about your research interests and how you ended up in this research thing so that people can have more idea about the profession, the research, and your everyday life.
Okay, maybe what I'll do right now, since there are a bunch of people in the lab, is show you around the lab and introduce you quickly to the projects. Then if you like, I can tell you a little bit about how this whole thing began. We have some brainstorming to also do with some of the teams, so you can also watch us, get a sense of the kind of thing that we do. Yeah, and then then we can talk about any questions that you have about the journey or anything else. Yeah. So let me introduce you all to everybody in the lab and what's going on here. We have two parts of the lab basically. We have a bunch of work done on crickets and bush crickets. Very few of whom are still, in fact, I think we've used them all. So we house them in these large sort of cages, which are semi-naturalistic. And then we do experiments measuring their responses to sounds. Here is the system that we use to sort of grow fish. That's our aqua fish system. So you can take a look at a bunch of housing racks that we have for zebrafish. Don't know if there's enough light for you to see the zebrafish themselves very well, but yeah, you can sort of see all of the zebrafish moving around in their individual tanks. We have multiple testing setups. This is one testing setup in the lab where there's a central tank, right? That tank over there in the center, and then display tanks, and the zebrafish see different social groups of different sizes and decide whom they would like to socialize with. And based on that, we get a sense of whether they have consistent preferences around socialization or not. This is where the zebrafish are bred. These are also various housing and holding tanks that we have for the fish. This is a setup that we use to measure two alternative force choice responses. So it's a T maze, right? So zebrafish would swim down this stem and make a decision to either go left or right, in that direction. So it's a T maze decision-making system. This is something called the novel tank test. The zebrafish are released in the middle, and then one sees whether they explore those two little objects which are new, those little plants or not. This is another T maze, but one that we're using, to explore optimism bias. So they learn to associate one side with a reward, another with a punishment, and then we look at mixed cues to see what their spontaneous response to mixed cues is, whether they assume that a mixed cue is indicative of something negative or something positive. This is also a setup where we look at neuroeconomic decision making, choices between two different sort of options. How do two quantities of two different objects sort of trade off against each other? And this is also where we grow zebrafish larvae and so on. I'm actually supposed to meet a group of students who are working on a particular project right now called the ecological value of depression. And so the group is investigating ways in which depression might or might not be, ecologically advantageous. And yeah, I think everybody's trying to not be caught up in the frame of camera. So yeah, I see that there are questions, so feel free to ask. And yeah. Go ahead, Sneha.
So, Professor, I'm sorry if this is a really ignorant question, but could you explain the utility of the research that you do on the crickets and the zebrafish and how that translates to psychology for human beings?
Well, so we don't do our work primarily to sort of focus directly on applicability to humans, but the applicability exists. So with bush crickets, we're studying the evolution of communication. We in general don't understand how human beings, how the evolution of human communication happened, and also how neurally speaking communication systems evolve. So that's the application of the cricket work, and the fish work is more narrowly clinically applicable because fish are vertebrates. There's a lot more similarity in systems. So the ecological value of depression, of course, has clinical value in terms of understanding depression as not necessarily being maladaptive, at least in the context in which it evolves. And so looking essentially at contexts where this might be a useful behavior and where there may be utility in terms of the signals being sent back. And likewise with the optimism bias. With all of the tasks that involve numerical cognition, we don't understand how human beings do numerical cognition. We need a good model system to ask those questions. So it's a very good question, and thank you for asking it. But you can feel free to ask about specific projects as well. All right. Anything else? All right. Well, if you want to observe more and not ask questions, and you can observe while we talk about one of our projects in the lab. This is the equal value depression sort of task. So do you want to just give an update, even though it's a recorded update about how things are going?
We put two fish for breeding,
Okay.
but they were taken from everywhere.
Well, you
One time.
yeah.
Yeah, so like we didn't know that the two tanks, which are labeled as male and female,
Yeah.
had both female fish.
Right.
As in like, because we bought it months back, we were very
Yeah.
small, we could identify them. But
Yeah.
just like, this is our first mate, we have bought medicine, we also consulted Nava.
Okay.
But right now that they have grown,
Yeah.
it's very, very evident that all of
The
them are
female.
females and
Okay.
so just one male.
Okay.
So I don't think we'll be able to like test the untested fish.
Yeah.
I think we just have to move on with the second set of trials
Okay.
for the pre-intervention stage.
Fine. That's okay.
That was just a major update that we have to give.
Okay.
And I think the last 3 files that we've tried to put for breeding, none of them have failed.
Okay.
So yeah, just those.
Okay.
We need to get.
How's the metabolic estimate coming along?
That's the thing. The battery was leaking inside the device. I checked it, and it's the battery is not working at all.
Okay.
I sent you the link to order the battery.
You just need a 9 volt, right?
Yeah.
Okay, fine. Okay. That's on the way.
It's mailed to.
Yeah, I've already added the parts.
And if we need to test more fish tests, we need to go get it again.
Yeah.
But oh, okay.
Then we have to go buy it.
Okay, cool. Because our existing batches are all utilized of fish.
Thank you. Not utilized, but most of them are females. So we can't.
Most of them are.
So we are limited by the.
Okay, cool. All right, so I guess we buy more fish. And I think the 9 volt battery should arrive by tomorrow. So then
You can start
check
with the
the
fish that have
metabolics.
already been tested
Okay.
for metabolic.
All right. Any luck finding possibilities in terms of us buying predators, Indian leaf fish availability? Will you look
Check
stuff
that.
up? Okay, do that. That'll be fun. Nice. Any other suggestions on experimental design that any of you have? Yeah.
I had one.
Yeah.
I talked to Pranay about the
Yeah.
other day, which was like there's some studies done in rats.
Yeah.
When they induce depression in them and they put it into a new environment.
Yeah.
They do not tend to explore it like the normal rats do.
Yeah, yeah.
So when we even put the fish
Yeah.
to
Acclimatize
record their movement. So there's an acclimatization period
Yeah.
that we give them.
That's right.
So I feel if we even record the movement in that period to kind
Yeah,
of see how
that's a good
they
idea.
actually
Yeah.
react to that.
And you can also use the novel tank test, the one that I just showed you, where there's a tank with novel objects. So you can see whether they're doing novel object exploration. We have the tank right here. I don't think anybody else is using it. So you can even go ahead. Yeah.
So that will be like an additional variable.
It will be an additional variable you're testing.
Yeah.
Yeah. Yeah. And are we settled on haloperidol then instead of resveratrol? Yeah, and reserpine, fine. So I'm buying the haloperidol then.
Okay.
You still haven't heard back about the things, right? But you will.
I can ask for this.
That's okay. Our own fund crunch has kind of eased. So worst case scenario, I can just cover it at my end. Cool. Any other updates? Okay. Cool. So who's volunteering to go get fish? Okay. Cool. All right.
But it's actually like one more person should go. Because it's
It's a lot to
because
carry,
I
yeah.
will get lost.
Yeah, that's true. But it's a fun
Yeah.
trip.
It's a fun trip.
It will be a fun.
it's an
All right. Cool. Any other updates?
It's the bad.
Yeah. Okay, cool. So I'll provide the battery, then we'll have metabolic readings by next week. And all of our fast fish, we won't have a post-intervention kind of. Actually, we can do a post-intervention even if they're all female.
Yeah.
Right? You just won't have enough to do.
Right.
You need another set of fish in order to
Just need to like, increase the samples.
Yeah. Okay, we have a question. Go ahead, Nirvik.
Hi, Professor. Thank you for such an engaging session. I
Hi.
wanted to, I wanted to ask whether there's any semblance or perception of other emotions amongst crickets, since you did mention depression and ecological footprint.
Well, we know that you can induce, actually both in zebrafish and in crickets, you can induce depression through what's called social defeat. So if pairs of crickets or pairs of zebrafish fight, then if they fight and one of them loses, then that induces depression-like behavior in both systems. Our other, I think depression is one of the most sort of identifiable other behaviors. And certainly, the opposite of depression you can sometimes see in organisms after they, for example, won in a fight or, after getting food. So zebrafish, for example, when are also more than food actually, really, really crave social interaction. So they will do many of the, in many of these tasks where it's assumed that you need to provide a sort of food reward, they actually sometimes function even better with a social reward, by which I mean they'll endlessly work in order to get a chance to just glimpse other fish. And so when they interact with other fish, they move a lot more. When they're isolated, they very quickly stop moving, stick to the side of the tank, and just show listless behavior. So in terms of that axis of sort of high movement and patterns of movement, you can certainly discern between different behavioral states in zebrafish. Hope that answered your
Thank you,
question.
Professor. Yeah, we have one more question in our chat. So Shraddha Joshi has asked, can neuroscience research help in artificial intelligence and vice versa?
Yes, it already has in some sense. Oh yeah, I see this in the chat box. So a lot of AI is built on what we know about neuroscience. So for example, how most AI are built off of neural network models. So they're based off of how we know neural networks to compute, which is that you have a representation of sensory input. Then you often have the best results in learning when you have an unconstrained middle layer of processing neurons with some plasticity in how they represent the information, and then you have an output. And the network can be trained on just comparisons of the output with either desired outcomes in, for example, untrained machine learning approaches, or some metric of performance in trained machine learning approaches. So in trained networks, you would provide some rules by which the network learns, but in untrained systems, you would just let some other metric of performance be the only feedback that the system gets. That overall structure is inspired by the brain, but also specific challenges in machine learning and in AI have come out of an understanding of how brains work. So, for example, a lot of visual image processing algorithms derive from how we know, neurons to do this. So you know that now sometimes when you're on Facebook, Facebook will suddenly start recognizing faces and tell you, is this so-and-so's face? Is this your friend's face? That algorithm comes from how we know brains to process visual information, quickly scan it for faces, zoom in on faces, and then identify faces. So that's an example of one of many things that where neuroscience research has helped with artificial intelligence. And vice versa, of course, artificial intelligence can sometimes, machine learning approaches can sometimes complement what I don't know if good examples where that's fed back to neuroscience research in the sense of directly in terms of what one wants to do with brains, but there are various things that brains don't do well, and you can use, you can use these systems, you can use AI to substitute for those. So, for example, a lot of medical diagnostics, I think, will soon have to be, thankfully, probably and hopefully make medical diagnostics more accessible by replacing doctors entirely with machine learning algorithms, because a doctor is diagnostically as good as the number of cases they've seen, whereas in a unified machine learning network, you can actually feed all of the medical data that's been digitized, and so the resulting diagnostician will have access to a much larger number of samples of data, a relatively objective diagnostic basis, and so on. So I think that will replace a lot of, thankfully, and reduce the subjectivity in a lot of diagnostic work medically. So there's several other examples like that I can think of, where AI will complement ways in which we know that there are limitations to the ways our brains compute in medical and scientific research. Yeah, go ahead.
Professor, thanks for coming in today.
Yeah, my pleasure.
Yes. So I was just wondering. So I had a shower thought, and so like, has studying neuroscience kind of like changed how you would define intelligence, and would you actually like qualify artificial intelligence as intelligence, or is there something about being alive, or like looking for something, being hungry, or being like wanting something that makes intelligence like different from, like, let's say a game of chess that a computer can play? Like, what are your thoughts on like intelligence in like general?
I think that I don't necessarily link intelligence to the necessity of being alive. I think that intelligence refers to a wider range of possible ways by which data and computation can be represented. So I don't necessarily link the two, but I do think that, of course, there may be qualitative differences to how different systems compute, and brains and living systems may compute differently from artificial systems. I'm also okay with ethical prioritization based on some degree of similarity. So prioritizing, say, the welfare of a living system over an artificially created system, and those are ethical questions without correct and wrong answers. But I certainly don't think of intelligence as being limited to living things. Yeah, go ahead, Sneha.
So then, Professor, talking about artificial systems in a different context, when we're talking about supposing conservation areas, right? So how do the ecological footprints and emotions as they relate to ecological footprints play out in those cases?
Sorry, I couldn't hear the first part of your question. Can you say that again?
Oh, I was thinking about sanctuaries and conservation areas, like breeding places. So stuff in places like that.
Places?
In breeding sanctuaries or conservation areas.
Yes.
So how do ecological footprints as they relate to emotions play out in those contexts? Because those are kind of artificially created systems as well, are they not?
They are absolutely. Yeah, they absolutely are, and many organisms don't flourish in artificial systems. And I think there's a wide understanding across the board that one wants to make these systems as natural-like as possible. We also try to do that. So, for example, we don't house insects in small sort of locations. We try and enlarge the areas that they have to live and breed, and hopefully we will increase the number of areas where we do this kind of thing as well. So yeah, that's that is the the hope is that more of us move towards just conserving larger portions of the world and not narrowly confining breeding sanctuaries to particular areas. Of course, they're necessary intervention when larger degrees of conservation are not happening and there isn't this clear lack of political will for larger degrees of conservation. breeding kind of sanctuaries are an intermediate solution, but they are far from being the ultimate solution.
Thank you for the answer, Professor.
Yeah. All right, go ahead, Shraddha.
So I had a simple question. How do you, as a neuroscientist, define consciousness?
Very good question. I sort of define consciousness in a somewhat limited way. So maybe a slightly boring definition relative to what could be the case. But I think of consciousness as the subset of the sensory world of which you are aware. And, you know, we are constantly processing constant sensory input, visual, auditory, you know, smells, touch, everything. I'm constantly, for example, being touched by my shirt, right? But I'm not constantly aware of being touched by my shirt. So I think of consciousness as a narrow sub-window of the sensory world to which we're paying attention. And not everything which we process is consciously processed. And so that's my unsatisfying answer. It's an attentional sub-window. And you know, is a perceptual, therefore also sub-window of the sensory world. Feel free to challenge that or tell me more. Yeah.
Apart from humans, does any other animals have this consciousness?
You know, the question is, how would you measure consciousness? And you can ask people that they consciously notice something and figure out whether they do or don't. Likewise, you can check whether animals have noticed something. Can they be trained to respond to it? So what we don't know is whether what we think of when we think of consciousness, we typically think of a whole range of complicated thoughts that we have. And we don't think of those thoughts as being restricted to a sensory sub-window of the world. So we don't know whether when another animal notices a sensory event, whether left to themselves, they're also recalling past sensory events consciously, whether they perceive the recollection of past events. We certainly know that they are that they're processing past events, that there can be memories of past events. All of those we know. We know there are neural tracings of all of them. But we don't know whether they have the same feeling, perceptual sort of sense that we do when we recollect something. So you don't know what the experience of being conscious is like for another organism. You know, we can just about get at the question of whether some events are perceived by animals and whether there are other events that are sensorily processed but not perceived by animals. And so certainly other animals have attentional sub-windows of the sensory world. But whether that experience is like what we think of as the human conscious experience, one can never quite tell.
Thank you for your answer.
Yeah.
Hi, Professor. So, does that mean that you said it is just, it's the experience that we have about consciousness, about what we experience, and it's very boxed. But so just a thought, does that mean that the evolution that humans have had, we're not the humans today, we are the blankets we use or the computer we use are not the humans we were even 200 years back. So in that sense, then the consciousness, then was it boxed back then too? So to what limit was it boxed back then, and to what limit is it boxed today? And then once the artificial intelligence, then we're seeing how the world is going. So to what extent will it be boxed in coming near future?
Yeah, I mean, hopefully our awareness of all the ways in which one can be conscious and compute and so on will, and have changed historically. What comes in the future is likely to be very, very interesting. But I don't think of consciousness as being, or the ability to compute as being restricted, as I said, to just natural systems. So without those restrictions, hopefully, whatever happens in the future, all the interesting stuff that happens in the future we will not be sort of unwilling to digest these new forms of data and forms of thinking, and hopefully we'll just be open to them.
Thank you, Professor.
Thank you, Professor, and thank you everyone who joined us tonight. So
Yeah.
our time is almost up, and if you have any lingering questions, I think we'll probably take one more question. So,
Yeah.
anyone?
So in that sense, just looking at it, philosophically, or just looking at it in a very social way. So in that sense, is it the thought? Because I'm not a student of neuroscience, and I might be wrong in saying this, but so is it that whatever, to some extent, we did as a civilization come to some point, and theories come up and philosophies come on and come up, keep on coming up, and the way of living keeps on coming up. So in that sense, is understanding of human being, just because we have had philosophers from the time of Aristotle about understanding of human nature and humans, and then today still we have people talking about it. So does that, in your definition, does that mean does that mean it's the same thing being repeated? Because if you go by that, it's a restricted sense of our understanding of the sensors that we feel outside us. So in your sense, it just means that it's the same thing being repeated all over again. The understanding.
Yeah, that's right. But all sensory, all brain activity is just that. But that doesn't mean that it's not complicated. We have loops after loops of cells that repeat, rehearse, and think about what decisions to make given very complicated aspects of sensory input. And sometimes some of these calculations we don't even respect as being complicated. So, for example, when you're deciding whether to go meet a friend or not, you are actually rehearsing large amounts of difficult to parse, low information data about how your past experiences with that friend were, what their smile looked like, whether you thought that they really wanted you to join them or didn't, whether you felt that something about the way they spoke indicated that you should call them back right away or call them back a little bit later to not seem too eager. These are complicated calculations. So our social interactions, for example, are very complicated calculations. And so, they are just all processed, rehashed sensory data. But that doesn't mean that it can't be complicated and interesting ways of processing data.
That's interesting. Thank you.
All very nice questions. And it's actually really lovely to interact with all of you and to engage with students who are so interested. So if any of you ever want to do anything in the lab or work here, just feel free to give us a shout out. Our lab's always open to anybody who'd like to intern here, as long as we can figure out the logistics for you. That we'd be very happy to have people in the lab interested in this kind of work.
Thank you, Professor. So it's a lab based in the university itself?
Yes, it's in the Ashoka University campus. Yeah.
Perfect. So if anyone wants to get in touch with Professor Bittu, I think you can reach out to the Kuhiro team. We will be able to connect you to Professor, provide you the email address and all information needed. And thank you everyone for joining us tonight. It was lovely.
Oh, sorry. It's just bittu@ashoka.edu.in. You can also find me on the website.
Yeah. Perfect. Yeah. Thank you, Professor, for your time and your effort. Hope it was fun for you as well to engage with such curious minds here.
Absolutely.
Yes. Thank you, everyone.
Thank you
We
all.
will have such further talks. Yeah. We'll